Nucleus or nucleoid. Contains genetic material, DNA and associated proteins. Nucleus is membrane enclosed. Plasma membrane. 1. Tough, flexible lipid bilayer. 2. Selectively permeable to polar substances. 3. Includes membrane proteins that function in transport, signal reception, and as enzymes. Cytoplasm. Aqueous cell contents and suspended particles and organelles. Phylogeny. The evolutionary development and history of a species or higher taxonomic grouping of organisms. How phylogeny trees are formed. Similar nucleotide sequences of the ribosomal RNAs of each group. More similar equals closer branches. Distances between branches equals degree difference between two sequences. Phototrophs. Two types: autotrophs, carbon from CO2, X, cyanobacteria; vascular plants. Heterotrophs, carbon from organic compounds, X, purple bacteria, green bacteria. Chemotrophs. Two types: lithotrophs, inorganic fuels, X, sulfur bacteria, hydrogen bacteria. Organotrophs, organic fuels, X, most bacteria, all non-phototrophic eukaryotes, humans. Prokaryotic cell structure. 1. Nucleoid. Contains a single, simple, long circular DNA molecule. There is no envelope containing DNA or specialized cells. 2. Ribosomes. Bacterial ribosomes are smaller than eukaryote ribosomes, but still synthesize protein from an RNA message. They are free-floating in prokaryotes. 3. Pili. Provides points of adhesion to surface of other cells. 4. Flagella. Propel cell through its surroundings. Eukaryotic cell structure. 1. Larger than bacteria. 2. Distinguishing characteristics: nucleus and membrane enclosed organelles with specific FXN, mitochondria, endoplasmic reticulum, Golgi complex, peroxisomes, lysosomes, vacuoles and chloroplast. Plant only. 3. Cytoplasm. Granules or droplets, starch and fat cytoplasm. 1. Highly dynamic structure. 2. Made of cytoskeleton, that is made of protein filaments that crisscrossing to make 3D network. 3. These protein subunits are noncovalently bonded to form filaments of uniform thickness. 4. Filaments are not permanent, they undergo constant disassembly into protein subunits, and reassembly into filaments. 5. Provide structure and organization to cytoplasm and shape to the cell. Name the 3 types of cytoskeletal filaments. 1. Actin filaments. 2. Stain labels. Microtubules. Fibers. 3. Intermediate microtubules. Green stain labels M, radiating from the cell center. 3. Intermediate filaments. Blue stain labels chromosomes in cells. Build hierarchical structures. 1. Nucleotides to DNA to chromatin to nucleus. 2. Amino acid to proteins to plasma membranes. 3. Sugars to cellulose to cell wall. Biochemical unity. All organisms share common evolutionary origin. Methyl, ethyl, phenyl, carbonyl, carboxylic, hydroxyl, enol, ether, acetyl, anhydride, amine, amide, amine, and substituted amine. Guanidinum, imidazole, sulfhydryl, disulfide, thioester, phosphoryl, phosphonhydride, mixed anhydride, What 6 characteristics distinguish living organisms from inanimate objects? ANs, living organisms, 1. Are chemically complex and highly organized, 2. Extract, transform, and use energy from their environment, 3. Have the capacity to precisely self-replicate and self-assemble, 4. Exploit a chemical interplay with their environment, 5. Possess programmatically defined functions, and 6. Evolve to new forms over many generations. All cells are surrounded by a plasma membrane composed of lipid and protein molecules. What is the function of the plasma membrane? ANs, the plasma membrane acts as a barrier to the free passage of inorganic ions and most other charged or polar compounds into a route of the cell. It contains proteins that can transport specific ions or molecules. Other membrane proteins act as receptors that transmit signals from the outside to the inside of the cell. E. coli is known as a gram-negative bacterial species. A. How is this determined? B. How do gram-negative bacteria differ structurally from gram-positive bacteria? ANs. A. Gram-negative bacteria have little affinity for the digestion violet used in grams stain, but gram-positive bacteria retain gram stain. B. Gram-negative bacteria have an outer membrane and a peptide O-glycan layer. Gram-positive bacteria lack an outer membrane and the peptide O-glycan layer is much thicker. Most cells of higher plants have a cell wall outside the plasma membrane. What is the function of the cell wall? The cell wall provides a rigid, protective shell for the cell. It is porous, allowing water and small molecules to pass readily, but it is rigid enough to resist the swelling of the cell caused by the accumulation of water. See Fig. 1-7, p. 6. List the types of noncovalent interactions that are important in providing stability to the three-dimensional structures of macromolecules. B. Why is it important that these interactions be noncovalent, rather than covalent, bonds? Noncovalent interactions include hydrogen bonds, ionic interactions between charged groups, van der Waals interactions, and hydrophobic interactions. B. Because noncovalent interactions are weak, they can form, break, and reform more rapidly and with less energy input than can covalent bonds. This is important to maintain the flexibility needed in macromolecules. Draw the structures of the following functional groups in their unionized forms. A. Hydroxyl. B. Carboxyl. C. Amino. D. Phosphoryl. ANs. What is the underlying, organizing biochemical principle that results in the chemical similarity of virtually all living things? Given this biochemical similarity, how is the structural and functional diversity of living things possible? Name two functions of proteins, nucleic acids, polysaccharides, lipids. Many answers are possible, including A. Proteins function as enzymes, structural elements, signal carriers, transporters. B. Nucleic acids store and transmit genetic information and act as both structural and catalytic elements. C. Polysaccharides serve as energy-yielding fuel stores and cellular and extracellular structural and recognition elements. D. Lipids function as membrane components, fuel stores, and cellular signals. Why is an asymmetric carbon atom called a chiral center? ANs, and asymmetric carbon has 4 different substituents attached, and cannot be superimposed on its mirror image, as a right hand cannot fit into a left glove. Thus a molecule with one chiral carbon will have 2 stereoisomers which may be distinguishable from one another in a biological differentiate between configuration and conformation. A. What is optical activity? B. How did Louis Pasteur arrive at an explanation for the phenomenon of optical activity? A. Optical activity is the capacity of a substance to rotate the plane of plane polarized light. B. Using fine forceps, he was able to separate the 2 types of crystals found in tartaric acid, racemic acid, that are identical in shape, but mirror images of each other. One sample rotated polarized light to the left, the mirror image crystals rotated polarized light to the right. A chemist working in a pharmaceutical lab synthesized a new drug as a racemic mixture. Why is it important that she separate the 2 enantiomers and test each for its biological activity? ANs, biomolecules such as receptors for drugs are stereospecific, so each of the 2 enantiomers of the drug may have very different effects on an organism. One may be beneficial, the other toxic, or one enantiomer may be ineffective and its presence could reduce the efficacy of the other enantiomer. Explain why living organisms are able to produce particular chiral forms of different biomolecules, while laboratory chemical synthesis usually produces a racemic mixture. ANs, laboratory syntheses usually use achiral reagents and thus produce racemic mixtures of products. In contrast, because all enzymes are made of chiral precursors, all enzymes are inherently chiral catalysts, thus they will show strong stereoselectivity in reactants and mechanisms leading to the production of chiral products. Proteins are constantly being synthesized in a living cell. Why doesn't the number of protein molecules become too great for the cell to contain, leading to cell destruction? Hence, the proteins in a cell are continuously being synthesized and degraded. The cell maintains a dynamic steady state in which the amount of each protein remains fairly constant at the level required under given conditions. Describe the relationship between a living organism and its surroundings in terms of both matter and energy. Hence, living organisms are open systems and exchange both matter and energy with their surroundings. They are not at equilibrium with their surroundings, that is, the concentrations of molecules inside the cells of the organism are not the same as their concentrations in the surroundings. To maintain this situation, the organism must acquire energy from its surroundings, either in the form of chemical energy or directly from sunlight. The free energy change for the formation of a protein from the individual amino acids is positive, and is thus an endergonic reaction. How, then, do cells accomplish this process? Hence, the endergonic, thermodynamically unfavorable reaction is coupled to an exergonic, thermodynamically favorable reaction through a shared intermediate, so that the overall free energy change of the coupled reactions is negative. The overall reaction is exergonic. Instant cold packs get cold when the contents, usually solid urea and liquid water, are mixed, producing an aqueous solution of urea. Although this process is clearly spontaneous, the products are colder than the reactants. Explain how this is possible in terms of the difference between dg. Hence, since the dissolution reaction is spontaneous, the dg must be negative. Since the reaction absorbs heat, the dH must be positive. Given dg equals dH, TdS, this is possible if the ds is very large and positive, as one would expect for a solid dissolving. On the reaction coordinate diagram shown below, label the transition state and the overall free energy change dg. For the uncatalyzed reaction ARBB, was this an exergonic or endergonic reaction? C. Draw a second curve showing the energetics of the reaction if it were. Hence, an exergonic reaction. What is meant by feedback inhibition and why is it important in a living organism? Hence, feedback inhibition is the regulation of a biochemical pathway in which a reaction product inhibits an earlier, usually the first step in the pathway. It is an important type of regulation because it ensures that energy is not wasted by an organism producing molecules it does not need. How is the genetic information encoded in DNA, and how is a new copy of DNA synthesized? And the genetic information is encoded in the linear sequence order of the 4 different deoxyribonucleotides in the DNA. When a new copy of DNA is needed, the 2 strands of the DNA unwind, and each strand serves as a template on which a new strand is synthesized. Hereditary transmission of genetic information can be viewed as a balance between stability and change. Explain. Discuss how a mutation in DNA could be harmful or beneficial to an organism. And some mutations lead to the synthesis of an inactive or defective enzyme or other protein that can no longer carry out its proper function, which is thus harmful to the organism. However, other mutations may lead to a more stable enzyme or to a protein that is better able to carry out its function in a particular environment, making it beneficial to the organism. Describe Stanley Miller's experiment and its relevance. Describe the RNA world hypothesis. Initially, RNA molecules were both genes and catalysts. Self-replication of these molecules over long periods of time produced variants that were able to catalyze polymerization of amino acids to form peptides that assumed the function of catalysts. Eventually, genomic RNA was copied into DNA, which assumed the function of genetic information storage. Describe how the rise of O2-producing bacteria might have led to the eventual predominance of Aerobic organisms on Earth. The rise of O2-producing bacteria would result in an increase in the levels of O2 in the Earth's atmosphere. This would give a selective advantage to aerobic organisms, which utilized O2 as electron acceptor, over anaerobic organisms for which O2 was toxic. What is meant by endosymbiotic association? How can this concept explain the evolution of eukaryotic cells that are capable of carrying out photosynthesis and aerobic metabolism?